great pleasure to be on this meeting again. Really insightful talks already, and looking forward to the next 1.5 days remaining. So, as it's been already discussed on this conference, long-read sequencing such as Oxford Nanopore really enhances our ability not just to identify structure variants, but also in terms of phasing, as we just heard from the previous talk, but also access our ability into repetitive regions such as the 386 medical variant genes that we published with Genome in a Bottle. So giving all these advantages and different aspects, we were of course also continuously looking into improving Sniffles going forward. And this is kind of the boring slides because it's just a single sample slides, but basically it works very well, as you can see here across different benchmarks from Genome in a Bottle and genome-wide or challenging medical relevant genes. We are continuously now improving Sniffles, not just for insertion and deletions, as you can see here, but also for very large events where we recently just had a genotype bug that we are correcting with the new release. And then we are also focusing more on complex structure variants, such as inversions and nested and adjacent structure variants going forward. The things I wanted to talk to you today about a little bit more is the population scale, and then later on the somatic structure variant part, which because I think this is sometimes overlooked, therefore there is a lot of power to identify new biology here, or even clinical spaces. So traditionally, how we do population-wide or trio-based structure variant calling is we identify structure variants on a pair sample basis, and then use tools such as Terrari, Survivor, Jasmine, or others to merge these individual VCF files together. Now we are up for the problem that we don't 100% know if a certain structure variant is actually present in sample A and B, because it might have been just called in A and failed some thresholds or other things in B, or is indeed not present, right? And so for that, after the merging, usually we have to do a re-genotyping, which again works on a per sample basis or on a BEM basis. You can use several tools for that. We are going to come out with a new tool called Campic from Adam English, which we are currently using in all of us. And then this is on a per sample basis, so we have to again merge. And then we are finally there, we're having a fully squared off, as we call it, fully genotyped VCF file, which is then really needed for subsequent analysis, being a Mendelian inheritance or violation of that, or in larger studies in terms of associations. So with Sniffles, we thought this is a little bit ridiculous since we have been through this exercise through a couple of thousands of samples already, and it's really not fun to do that. So what we have come up with in Sniffles is to actually store all the candidate SVs, even if they are just reported by one single read, in a binary file that comes out of Sniffles if you add the tag -SNF. And this binary file is typically on the size of 12 to 20, 30 MB, so it's very small and manageable, right? And then when we invoke Sniffles merging operations, which is a separate parameter, you can feed in all the SNF files that you have, and that generates automatically a fully genotyped VCF file. So you don't have to go back and forth. The other thing which is really cool, you can keep these SNF files around and put the BEM files in cold storage, for example. And when you have new samples in addition to your population, you can just add them in the SNF file and do the merge again. And you again obtain a fully genotyped VCF file. This is significantly faster. The previous old approach that I just told you with QtSV and Survivor and so on and so forth, this takes around 36 CPU hours on a single trio. However, our approach just takes 65 seconds on the same data set, right? So there's lots of greatness about this, I think. Besides single sample calling. So we of course tested this out, not just over benchmarks, which I'm skipping right now, but also in real Mendelian samples. So we sequenced 20 or 30 Mendelian trios around the MACP2 disease together with Claudia to analyze how good Sniffles is in Mendelian consistency, but also how good or bad we can resolve these highly complex variants, right? And in short answer, it worked really nicely. 90% of all the Mendelian trios, we could find the disease, and we could resolve the disease-causing SV, no problem at all. To enhance this further, what my postdoc Xing Shang has been up to over the last 1 year or so is to work with Ryan Lehr from Colorado on STIX. STIX is a new form of annotating population frequencies for structure variants. Because the short thing of it is short reads are not great in structure variant calling, but they're even worse in structure variant population frequency annotation, believe it or not. And so what you can see here, we benchmarked this. So for HG002, that's the genome in a bottle structure variants, the benchmark itself. Gnomite, for example, can annotate 32% our tool is able to identify and annotate 85%. And so the tool currently operates on a set of 1,100 genomes from 1,000 Genomes project, from Danny Miller's, but also from the cohort from Austria that was sequenced in Austria. And the preprint will be out shortly. There are still challenges remaining, which is great because that keeps us going and keeps my job going, which is, for example, this scenario here, where we have quite a tricky region. So in orange, you see segmental duplications. On the top, you see data from a copy number array. And you see that actually in red, you have abnormal copies of that proband in this region itself. So what is actually going on, indicated by the arrows here, the red and blue arrows, is you have different segments of this copy number. In the segmental duplications in orange, you don't see any data from the array, of course. And the actual allele is actually displayed on the bottom of the page in that proband, right? So like what we have is basically a duplication event. And inside of the duplication event, we have a triplication that is also inverted simultaneously. Right now, Sniffles cannot report the accurate allele of this. Right now, Sniffles would report a de novo variant in terms of an inversion, but not resolve the entire allele itself. And this is part of it because we are lacking language in terms of a VCF file language, but part of it is also, it's just very complex to get these things right and get that generally resolved, but we are working on this. For cancer studies, Sniffles detects mosaic and somatic mutations. There was just a talk today morning. We solved that bug, so it should be better and come out in the next month or so with the next release. But this is pretty straightforward, and within seconds, and using this merging tool that I just described to you, this is now very straightforward to identify the somatic variants. So we are applying this to Gregor, all of us, and also working together with G42, which I think are now M42. They're changing their name. So this is great. The other thing, which is grabbing my passion, quite frankly, a little bit more these days, and I hope I don't run out of time because the timer isn't showing anything, is somatic structure variants. What I mean with them is really low frequency within the tissue. So you might heard of like tumor normal comparisons, right, where we have like mutations in a certain tissue, but maybe at high prevalence. But I'm really talking about low frequency mutations within the tissue, where you really have to go down into the signal versus noise and identify what you can really call a grasp. And so over the Somatic and Mosaic Consortium, SMAD, we have done now benchmarks with that, of calling structure variants down to like 2% or 5% variant allele frequency, or even below that. This also depends on your coverage that you have. And for example, we have benchmarked that Sniffles currently has a precision of 85% of structure variants down to 1%. And has a recall of like 10% of SVs that we are missing in these data sets. So I'm pretty happy with this. And this figure here actually sparked our idea back then. So what is happening here is on the left side, you have an ALU-Y element, and then you see the insertion, which is actually a germline insertion of another ALU-Y element, right? And then this little, I hope you can see this little black lines that Sniffles calls as a deletion, is actually a mosaic repeat recombinant that we can now call automatically, even on 30x nanopore data. This is not a base calling deletion error. This is actually a real event, and we wet lab validated it on the side. You see APC as Sangers, and then did also some Sanger sequencing on the bar chart here down here. And so in total, actually, in this one brain sample, we identified 26 ALU-ALUrecombinants in 50x data, sorry, and 125 insertion versus mosaic deletions in close proximity. So this is great fun for me, and we are really feeling passionate, and we are going forward with that, identifying also methylation impact of these mosaic SVs in these regions, and so maybe that's the next talk at Nanopore conference. But what we had fun with today, and I want to share this with you, is that population frequency tool that we just showed, right? So it's cool to look at, are these variants actually shared across individuals or not, right? So the germline SV, the germline insertion, is present in 53% of the 1,000 Genomes project. We did that with our genotyping tool. So now the cool thing comes, right? You have now two ALU elements that are close to each other in high prevalence in the population. And so the mosaic deletion, the mosaic recombinant, is actually present in around 3% of the individuals. This hints or highlights the possibility of independent gain mosaic mutations through our human population, which is really fun and cool, and like there's going to be a preprint in the next week or two. So to wrap things up, we are of course working together with CART doing a lot of brain sequencing in Oxford Nanopore and studying not just germline mutations or somatic mutations in different regions of the brain, but also mosaic mutations inside of the brain, which might also highlight different cell types itself. We're also doing this with cancer genome projects, such as the Michael Smith Genome Sequencing Center, where we just had an accepted paper in cell genomics, and then working also with SMART and genomics England a little bit. And with that, I think my time is up. I want to thank my entire lab, HGSC, the Human Genome Sequencing Center, UCL, and PNRI for great collaborations throughout the years. And then on the right side is some funding information. Thank you very much.